Good morning, Austria. Good afternoon, China. We are here at the EASD Miss China Daily Highlights in Vienna at the 61st EASD annual meeting. Today, with the great honor, we have Professor Raphael Scharfmann, the 19th Albert Renold Lecture winner with us. Professor Scharfmann, warm welcome.
It's very nice to be here with you in Vienna.
Thank you. Professor, we know as you are winning this Albert Renold Lecture, which is a prestigious reward, recognize the work, the good work you do. And we wonder, can you share with us what motivates you to do all the work, all the good work? And can you share with us your feeling about winning this award? Thank you.
So, first, it's very nice to receive an award from people that selected my application to get it, and I know it is people working in this field. I know it is very smart scientists, and it's very nice that the work I've been doing during the past maybe 30 years has been highlighted to receive this award.
Yeah.
What we have been doing during the past 30 years is to try to understand how pancreatic islet cells, the cells that make insulin, are produced and how they secrete insulin at the right time to regulate glycemia. And it's very nice to get awarded for what we have been doing during the past many years.
Thank you. And Professor, we know your team has successfully developed the world's first functional human bed cell line, which is a milestone achievement. Could you share with us maybe the major challenges faced and the innovation during the research process? And share also with us your views on the clinical impact of this finding on diabetes treatment.
Okay, so this is indeed a real achievement, and this is something we started to discuss long time ago. That means at the end of the early 2000s, and in fact it took us 12 years to publish our first paper on this topic.
Okay.
So it was extremely time consuming. It was a very difficult project, but we reached our objectives. That was to make the first functional human insulin producing cell line. And this cell line has been shared with many labs in the world, and we are extremely happy that now many labs work with this cell line that is not something to be used for cell therapy, but it is something that is very important to discover new molecules that can amplify insulin secretion, that can protect the cells against destruction that takes place in type 1 diabetes, that can protect cells that dysfunction that takes place in type 2 diabetes. So it's an object that, in my opinion, that will be extremely useful, and you can get them in large quantity to progress in our knowledge on what is a beta cell, what is its dysfunction in type 2 diabetes, and how it is destroyed in type 1 diabetes.
Thank you very much, Professor. And we also know that your team recently proposed the fusion model of islet formation in the Journal of Diabetes, which present a significant maybe challenge to the traditional diffusion model, right? So, which based on the mouse study. So do you think this innovative framework highlights the inherent limitation of a mouse model is stimulating human diabetes?
So the first important point is what we know on pancreas development has been mainly developed in rodent model, and this was extremely important. And I think we should continue to put effort on using mice to understand what is a beta cell, how the pancreas develops, etc., etc.
Yeah.
But it is also important to keep in mind that a human is not a mouse. There are many similarities, but also some differences.
Exactly.
And one of the differences we recently discovered by studying human pancreatic development is the fact that while in mice at the end of gestation, you have very big islets that are cut in pieces to give rise to the different islets we see in an adult mouse pancreas. It's a fission process. In human, it seems to be a bit different. It seems that islets first appear as very small clusters that will give rise to bigger clusters by fusion of small clusters to give rise to those clusters. I don't know whether it's important for function, but it is something we discovered and that might have some relevance that will appear in the future. So what is important, I think, is continue to make beautiful description in what is going on in different models to be able at the end to reconstruct what is a human islet.
Thank you, Professor. So I have a follow-up question based on what you all just shared excellently. So all the scientific work you do, so it is hopefully to translate into practice, clinical practice, right? So do you envisage the fusion model will open up new avenue or pathways for understanding diabetes pathogenesis and provide more targeted theoretical support for clinical treatment strategies?
So this is a bit difficult to answer at this stage because what we are discussing now is something really new, and you know that islets are composed of beta cells that produce and secrete insulin, but also you have other cell types, alpha cells and delta cells, and how beta cells interact with alpha and delta cells to regulate glycemia is a bit unclear at this stage. I think that putting on the table all new discoveries on how islets are budding, how they form by fusion or by fission, maybe in the future will help to understand how we can regulate glycemia in diabetic patients, either type 1 or type 2, in a tightly regulated manner.
Thank you, Professor. I have a final question, also based on what you just shared. For your work, you took decades, right? You mentioned 12 years, and you shared your excellent data result with different labs. So I wonder, in terms of this collaboration, international collaboration, particularly like we are from China, you are from France. Yesterday we have Professor Steve Kahn from the US. He also mentioned like scientific work without boundaries. So how do you see international collaboration among different countries globally? What is your view about this? And what is your future plan for your research? Thank you.
So the first point is, in the past we had quite a number of PhD students or postdocs that came from China to learn what was done in our countries, and many came back to China, and I have been impressed by the current quality of the Chinese science and what I can read in the scientific newspaper. I think that many progress have been done there, and I think it would be nice now I think that we French scientists would learn a lot for our labs by now interacting with Chinese labs, and maybe what we have done by translating our knowledge to China could be back to our European labs, and I would be extremely pleased to interact more with Chinese labs on the way we are doing science here, on the way people are doing science in China, how we translate our knowledge from academic labs to biotech companies and to pharma companies. And I think that putting everything together could make a strong plus to the science and to new strategy to treat and cure diabetes.
Thank you very much, Professor, for your sharing. So I'd like to conclude today's interview by saying thank you very much for your time and attention. Thank you. Bye-bye.
Bye-bye.